Okay, I hope. Okay, thank you for joining this talk. I'm delighted to have this opportunity to present our work, which is focused on defining individual variation in human pluripotent stem cells during their early lineage specification. So many previous studies have defined the various genetic and epigenetic sources of sources driving variation in multiple pluripotent stem cell lines. Despite this effort, we still lack a systematic assay to dissect and interpret variation in pluripotency. So in this study, we aim to address this topic by defining how the individual variation in pluripotent stem cell is functionally linked to early lineage diversification. Okay. So when single cell population of a pluripotent cell is plated and cultured as a monolayer, distinct cell states emerge in temporally and spatially organized way. These distinct states display different sensitivity to signaling molecules, as shown here with BMP4 treatment. When cells were treated with BMP4, expression of mesenteric fate regulator, CDX2 and brachyury was induced. When the BMP4 was treated on day 2, only cells at the age of the colonies were responsive to BMP4. On the other hand, when cells were treated with noginesb to induce neuroectoderm, we found SOX21 was induced at the core of the colonies. This shows that these distinct zones of pluripotent cell epitheliums are related to distinct lineage bias, showing mesenteric bias at the age and neuroectoderm bias at the core of the colonies. So this space organization has not been systematically tested across different lines previously. Here we took 6 cell lines and grew them in self-renewal and neuroectoderm condition. We looked at SOX21 and NANOG expression and found different lines show different levels of SOX21. SN01 lines show the highest and IO4 lines show the lowest level. So this simple assay showed that SOX21 expression level could be used as an indicator of neuroectodermal phase specification at the early times of differentiation. And these images at lower magnification make it easy to see the different efficiency in generating neuroectoderm between the two extreme lines, SA1 and IO4. And this difference was maintained in a certain range of concentration of neuroectodermal inducers and different plating densities. And if you remember Tyler's talk, here I think we have that SA01 shows actually dose-responsive effect with this treatment, but here we have IO4 which actually doesn't have a response to this given concentration that we tested. So we actually believe that for this line, I think we might need to intervene other signaling. So next we performed RNA sequencing analysis on the cells collected 2, 4, 6 days across 3 conditions for all 6 cell lines. Principal component analysis showed that PC1 reveals major gene expression changes was associated with the mesenteric differentiation triggered by BMP4 treatment. And PC2 showed progressive changes during the differentiation times for all the conditions. Although all 6 cell lines moved along similar trajectory in this space, we can see cell line differences are also evident along the differentiation trajectories. So next, we applied non-negative matrix vectorization method, which we called GW COGAPS analysis. And we acquired 22 transcriptomic patterns. Here, the culture days are shown on the x-axis. 6 cell lines are shown in the different symbols, and 3 conditions are in different colors. So through this analysis, two classes of pattern were identified. One class we describe as a dynamic pattern, define the transcriptomic changes that changes over cultural days or under different conditions, and were conserved among different lines. In contrast, the other class defines unique transcriptomic signature specific to each cell line, but unchanged across time and condition. And we describe these patterns as a cell line specific pattern. So here I'm showing 4 examples of our dynamic patterns. We projected the mouse gastrula single-cell RNA-seq data into our dynamic pattern, and this revealed that these transcriptomic modules define the reduction of pluripotent states during differentiation and induction of mesendoderm and neuroectoderm by BMP4 and Noginesib treatment. So here is shown how gene expression data can be defined by GW COGAPS analysis. In a conventional clustering analysis, gene expression pattern of these 3 genes shown in the middle would be defined by 3 patterns, and each gene expression will be explained by only one pattern. By GW COGAPS analysis, this gene expression data can be defined by 2 parents, and this method can assign contribution value of all the genes for each parent. So in this way, gene A would have a high contribution value to parent 1, and gene B would have a high contribution value to both parent 1 and 2. And this approach especially allows us to understand the multiple use of a single gene for multiple biological events or function. So applying this method, OTX21 gene expression in our data was decomposed by one dynamic pattern showing induction in Noginesib condition and another pattern specific to UCO6 cell line. And here the numbers show the gene contribution weight values, which are the gene weight of OTX1 for each of these 2 patterns. So these cell line specific patterns were particularly interesting for us, given our focus on defining cell line variation. These patterns identify transcriptomic signature that were unchanged across time and condition, but show the higher levels in specific cell lines. Here, I'm showing the H9 specific pattern as an example. And when we project embryo body-based differentiation data into this H9 specific pattern, it successfully distinguished the H9 sample from all the other lines. Additionally, when RNA-seq data from other group was projected to this H9 specific pattern, their H9 sample showed the strongest signal. This suggests that individual lines have unique transcriptional traits, and we can successfully define this by GW COGAPS analysis. Interestingly, we found that this cell line specific signature can be used to predict the differentiation potential of the line. Retinoic acid signaling plays a key role in hindbrain formation in early development. And we assessed the enrichment of 252 retinoic acid responsive gene in the line specific pattern. And line-specific pattern of IO4, which show low efficiency in generating neuroectoderm, show the higher retinoic acid gene enrichment compared to SAO1, the line that showed the higher efficiency in generating neuroectoderm. To further validate this finding, we tested actual retinoic acid response and hindbrain differentiation efficiency by monitoring the HOX-B1 induction after retinoic acid treatment. Here, line IO4713 exhibited a higher response to retinoic acid compared to line SA1 IO4-UCO6. And we also found that there is a strong correlation between retinoic acid gene enrichment in the line-specific pattern and the efficiency in the generating hindbrain fate. Next, we tested this retinoic acid response on further neural differentiation, and IO4 line generated more hindbrain neuron than SAO1. And all these findings demonstrate the predictive power of cell line specific signature for the differentiation potential of individual line. So in addition to the finding of this cell line specific pattern, we also noticed that SOX21 shows its highest gene weight in one of the dynamic neuroectodermal pattern, P15. Moreover, SOX21 shows the slightly higher gene weight in SA1 line specific pattern compared to IO4 line specific pattern. And because the role of SOX21 in the early specification has not been previously described, we investigated its function using knockout line generated from SA1 line. So here I'm just presenting one of the key findings showing the induction of brachyury and CDX2 in the SOX21 knockout line under BMP4 treated on day 2. And briefly, we found the SOX21 induces anterior neuroectoderm by inhibiting mesendoderm and neuromesodermal specification. And consistent with these results, we also found that knockout of SOX21 in SAO1 increases its retinoic acid response, resulting in higher efficiency in generating hindbrain fate. So altogether, all these findings suggest that both dynamic and cell line specific pattern can be used to assess the specific signaling response and lineage bias of specific cell lines. And to further validate our approach, we generated new set of iPSC lines from fibroblasts of 3 donors. The RNA samples were collected from 2 replicates like from each genome. And GW COGAPS analysis of this new RNA-seq data again revealed the donor-specific pattern that show unique transcriptional signature for each donors. Then we projected RNA-seq data from the cerebral cortex of 260 individuals into the donor-specific pattern that we defined using pluripotent cell data. And this demonstrates that these donor-specific signatures were also present in the corresponding donor's adult tissue. The projection of parental fibroblast data into the donor-specific pattern also distinguishes the corresponding donor. And this data indicates that donor-specific signatures are stable traits of an individual human, persisting in different cells throughout their life. So to further investigate lineage variation in extensive collection of lines derived from the wider human population, we explored the published RNA-seq data collected from 300 undifferentiated iPS cells derived from 100 donors generated by Next-Gen Consortium. In this PCA plot, cell lines from each donors are arranged along the x-axis based on their PC1 level. And each lines are colored by its average expression level of retinoic acid gene, with the higher level shown in green. This analysis showed that PC1 exhibited a strong positive correlation with the retinoic gene expression, while PC2 showed a negative correlation. Next, we projected mouse and macaque gastrula data into this PC. And high PC1 values were associated with mesoderm and endodermal identity, shown in yellow to red in here. And on the other hand, High PC2 values were associated with epiblast, primitive streak, and primitive erythroid identities. And this correlation confirms that variation in retinoic acid response and early lineage bias that we observed in a small number of lines previously are indeed a major variation in multiple lines derived from the broader human population. So here, PC1 and PC2 of next-gen data are plotted together. And retinoic gene expression level in each cell line are colored. This strong association with retinoic gene expression and early lineage bias were further validated by projected mouse embryo data generated by laser capture microdissection. And the PC3 of this data showed transcriptomic difference in anterior to posterior regional identity of the embryo. By projecting this next-gen data, we observed a strong correlation between the anterior bias in lines with the low PC1 values and the posterior bias in lines with the high PC1 values. And this finding indicates that transcriptomic variation in pluripotent cell line align with the developmental anterior to posterior axis. And furthermore, we found that next-gen PC2 is correlated with a mouse embryonic stem cell versus mouse epiblast stem cell identity. The cell line with the high next-gen PC2 values display the highest levels of mouse embryonic stem cell identities. And last, we projected our initial 6 cell line data into the next-gen PC space. And here, I want to remind you that our 6 cell line data include early differentiation data triggered by Nogin SB and BMP for treatment, while only undifferentiation data define the next-gen PC space. Therefore, through this projection, we can test how the variation in undifferentiated cell in next-gen data is correlated with the early specification. And this projection clearly separated self-renewal neuroectodome from mesendodome differentiation along the next-gen PC1. And temporal progression of self-renewal towards neuroectodome and mesendodome was aligned with the PC2. And here you can also see the remarkable similarity between the two PC space defined by the 6 cell line data and next-gen data. And all this analysis confirm that lineage bias along the anterior to posterior axis is correlated with the transcriptomic variation in undifferentiated state and allows us to assess lineage bias systematically for an extensive collection of pluripotent cell lines. So in summary, our study showed that functional variation in early phase across different lines can be defined by formation of developmentally existing zones. We showed transcriptional variation between the lines can be defined by dynamic and cell line specific transcriptional signature. And we also demonstrated cell line specific signature can be used to predict specific signaling responses and differentiation bias of individual line. And finally, we show variation in lineage bias across large collection of pluripotent cell lines aligned with the anterior to posterior identity, retinoic acid response, and lineage specification. And we believe these early cellular and transcriptomic assay using pluripotency and early cell stage have the potential to predict and assess the new feature of genetic and epigenetic bias across the extensive pluripotent cell line collection. So this work is done with a huge collaborative work from many teams and members shown here. And the project was initially started and performed mainly at Liver Institute in Baltimore and continued by collaboration with another Sestan lab at Yale University. Okay, and thank you for listening, and I will take a comment and question from here.
Thanks, Suel-Kee. Any questions?
I want to say that was such a fantastic presentation. Thank you so much. And you saw what Taylor presented, that we see these donor-to-donor variants. I think that it's really interesting to dig into to what extent this is truly donor specific versus that IPS lines can take a few different states and then, you know, that there's a sampling thing that shows is a donor to donor. So my question is, what is the sort of biggest number of lines you have per donor that really indicates to you this is a donor specific effect?
That great question is actually quite difficult to answer. Okay, so for the donor specific, okay, so the donor specific pattern that we actually identified shows quite a stable trait that we see actually see the trait from all the different states. And I think that's very, contribute with the genetic factor. And we actually did several analysis, and then one of the findings is actually this donor specific pattern is more enriched with the evolutionary newer gene, and also they are kind of show the very subtle differentiation. When we actually use the COGAPS analysis to identify this cell line specific pattern, here actually there is a specific condition that we triggered. So there is a login SB and BMP4 condition, which mainly we actually perturb the BMP4 signaling in the system, right? And then I think we dissecting out the BMP4 response as a dynamic pattern, and also some of other very stable trait, which is linked with donor-specific feature, as separated out with as a cell line specific pattern. And actually, one of the analysis that I didn't show here is when you actually did a MDS analysis with this all the pattern that we identified, you can see these are the BMP4 pattern, and these are the neuroectodermal pattern, and these are the cell line specific pattern that we identified. These are separated from the dynamic pattern in MDS dimension 1. But interestingly, we can see that the some of the donor space pattern are actually close to the BMP4 pattern, and the other cells, which actually make a more neuroectoderm, are more close to neuroectoderm pattern. So dimension 2 is actually showing the lineage bias and how this donor specific pattern and dynamic pattern are intertwined. I don't know how to express that. And because of there are some features, the donor specific pattern has linked with lineage bias. That's why I think we are seeing these retinol acid responses in the donor specific pattern.
Okay.
So I think right now we actually played with only 6 cell line. We have many different lines. How we can actually define the donor specific pattern, individual donor specific pattern, it's something that we need to explore more. But when we actually do the connectivity analysis, we can actually we with many analyses, we actually separate out donor or donor which shows similar characters with the other individual. And that's something we actually see some level with, sorry to move around, where is it? Here, that here, which is a 3 donor specific pattern that we identified. When you project it, we point out this donor, but among many donors, there are many donors which is more close to this particular donor. So I think this is really, I mean, Sally, what you ask is a really interesting question, and we are actually trying to develop more clear say for this, to measure all this stuff. Hopefully I answered the question well.
Yeah, that's beautiful. Thank you.
Yeah, and in the next gen data, they had 101 donors with 317 lines. So somewhere between 2 and 5 or 6 lines per donor. So there was also, whatever dimension you're looking at, whether it was a PC in that data, a single gene, or a projection of a signal, you can assess from that larger collection how much variation is inside a set of 5 lines from one donor versus the variation across the entire population. So depending on what you want to ask, some of the large collections can answer those kinds of questions.
And Carlo, do those include lines that have gone through CRISPR, the CRISPR process?
There's no. The next gen consortium is just all lines, no editing. Although Suel-Kee has the SAO1 SOX21 knockout, so we could have a singleton to look at there, but we haven't systematically looked at edited cell lines.
Yeah, or unedited cell lines, right?
Right.
The line still goes through the process, but it's not edited. Yeah.
Gotcha. Right.
Those are the types of lines we use, right, in our studies. So I feel like sometimes there's a disconnect. People are looking at a lot of parent lines for these sorts of studies, but then when we go to use the protocol, we're using a whole different line. So if that CRISPR process is doing something, yeah, be good to include those.
Can I just make a comment just to reinforce you? That's really, there's so many different sources of variation. I wasn't expecting you to say that just now. That's right. You could pick that up too if you wanted. And you probably will have to. But that's what's so beautiful about the field at the moment, is that these tools are getting precise enough that we're going to see this variation over and over again.
Yeah.
It's getting real. And but Sally, your question, it seems to me, I mean, if you looked at that image where we take that's Ihor Lemishka's next gen data we're talking about here. It's very early, right? 100 genomes, right? Let's say in triplicate, roughly triplicate. Some are 5, some are 2s. That PC1 there, that's genetic. That's what people mean when they say genetic, right? And it's a continuous.
Yeah,
so it's possible that there are states, just as you and Tyler were discussing, that are also present in this system. And they may be related to this, I don't know. But it's very interesting, right? Am I being clear? I think then when you start doing the projections into the mouse and macaque gastrula, right, you see, particularly if you go to the next slide where you take the UMAP data, UMAP 1 and 2 from the next gen PC1, right? That's the population. That's 100 humans, right? And they occupy all these different states. So, for example, I mean, we could go now and look at TBR1 or any other gene you're interested in, and see whether it varies in this population. And when it does, it varies systematically. That's the point. It's not just random. It seems to map the human embryo.
Mm-hmm.
Right? And then you think, well, is that what you'd expect? And sort of it is, but then that's what we want to study. Right? How does this variation influence gastrulation?
So
I don't know if
yeah,
that's helpful.
So I think
It
that's
is
how
helpful. It is helpful. I would just say that it would be really interesting to make more lines from a particular donor, to make those lines in different ways, to grow them in different media.
Oh
you
yeah,
know,
sure.
so that when you've got a lot of lines, because if you only have maybe 1 to 3 lines per donor, which is what people often do, then you know, you worry that the particular way those lines were made
Right.
can
So let,
bias your
Sally, let me
viewpoint.
respond also
That's all.
here, not to be too interventionist, but we do see that too, right?
Okay.
And Suel-Kee didn't emphasize it here, but from individual donors, we see lines with quite distinct properties of this retinoic acid response type that we've characterized so well.
within donors.
So you see that too. And then this game gets very, very interesting, because then that's highly stable as well. So that's epigenetic. Yeah, this gets interesting. I hope that everyone who's on board still is kind of enjoying this. We could explore it in any way you want, but one way or another, it has to be explored. It's not just some arcane sideline by people who don't have anything better to do.
Alex Jordan just sent in a question saying, do you think this is specific to the AP axis, or could this occur across other axes, dorsal ventral, medial lateral? And I think all of us would say, yeah, this isn't just AP. The question is, how many of these axes could we track human variation across at this early time? And I think most of us would agree that there's many here to be mapped. There's a lot going on in pluripotency.
Well, Carlo,
Yeah,
dorsoventral specification was demonstrated in our work, right? Michele et al. 2020.
yes, at a later step, but yes, for
Right.
sure.
It's a later step, but it shows
So,
you know, dorsoventral bias of the same line. So I think it is anteroventral versus dorsocaudal bias, right?
Right. Anything these
Or-
people are going to
yeah,
have to
those-
do,
right, I
different
think
humans can be
that's
different.
the answer that we can give Jordan. I think, yes. So the anterior biased line, actually, we found that they are more biased to ventral identity. And then posterior neuroectodermal biased line, actually, we found that they are more biased to dorsal right, the dorsal identity, right? So there was actually link with the early anterior posterior axis
Right. So
to
right.
later dorsal ventral axis, and that's actually published in separate paper in our study. But anyway, that's yes, there we saw the link, but we couldn't do it with the Lemesca data here because it's undifferentiation data. There was anterior posterior bias that we can capture in this data, but we couldn't see actually dorsal ventral bias in this particular data.
Because it's a later feature, right?
Yes.
Actually, it's interesting how the axis, the developmental axis, are recapitulated by this progression with the iPS cells, right? Anteroposterior axis early, and then dorsoventral axis later with the neural stem cells. Once they are neural stem cells, yeah.
Yeah. So this issue of timing that we're discussing is really critical, right? It's how the system works, right? That the events are timed. That's what's so extraordinary about it. And yeah, I mean, I think not to sound too philosophical, but the point I really want to make about this discussion you were just having is that the only way to find out is to look at the cells. And that sounds as if it's very sort of difficult to do, but actually all these single cell tools that are being developed are getting to the level where we should be able to see in some depth the origins of these different biases right at the beginning, and see whether they do in fact play out at different points in the cascade.
A note on
I mean, the key is to understand how organizers work. An organizer is a dynamic cell interaction that's taking place very quickly to specify cell type. Right? That's what's going on.
Yeah, and maybe to